Hello and welcome to a video on cofactors, specifically looking at coenzymes, prosthetic groups and inorganic ions. So these coenzymes, prosthetic groups, inorganic ions are types of cofactors. So it's like a cofactor is the overarching branch of all these smaller components. So a cofactor is any substance that has to be present to ensure that an enzyme catalyzed reaction takes place at an appropriate rate. So this is a substance that's necessary for enzyme reaction. And there's lots of different types of them. So we've got our prosthetic groups, and they're non-protein parts. So I'll do this in red actually. So the non-protein but they are integral to the shape of the enzyme. So if we've got a tertiary structure, then the prosthetic group would be integral to the structure and it wouldn't have the same shape if it didn't have this prosthetic group. So an example of that is carbonic anhydrase, which is an enzyme. And it contains a zinc ion that is permanently bound to its active site, and they're found in red blood cells or erythrocytes, and they catalyze reactions of carbon dioxide and water to carbonic acid. So again, they're just integral to catalysing the reaction that enzyme does or catalyses. So without it, we wouldn't be able to make the products that we need. So that's this one. There's lots of different examples of them, but that's just one. So we need the zinc ion that's permanently bound to the active site of carbonic anhydrase in order to allow these reactions to take place. If we look at coenzymes, they are organic non-protein, again, so they're not non-protein molecule that bind temporarily though. So whereas these are permanent, these are only temporary binding. Bind to the active site of the enzymes. So, for example, it could be folic acid, or any kind of vitamin, really. They are needed in the body. Right. So without the folic acid, we can have diseases formed, so like human deficiency. So if we have a lack of folic acid, it might result in anemia. And that's just one example, but there are quite a lot of examples for that. So that's a coenzyme. So you need to digest the folic acid. And then we can look at inorganic ions. So an inorganic ion is a temporary change. Also for these, they are chemically changed, so another enzyme will need to change the shape of the active site. So anyway, inorganic ions they're temporally changed, and so they can attract either substrate or active site. So an example of this is that the reaction of starch to maltose uses the enzyme amylase, but the reaction won't be able to take place if we didn't have chloride ions. So that's just another example of why they are integral to that reaction. And lastly, we have, this is only a smaller part really, but I just wanted to add this in. We can have co-substrates, so we can see here that this enzyme with the active site here, both of these substrates can bind together and bind to the active site, so on their own they won't be complementary, but together they are. So that they are the complementary shape for the active site of the enzyme, so they can bind and form an enzyme substrate complex. Thank you for watching.